Hi, everyone. We're just giving it 1 minute or so to allow people to enter into the Zoomovere. I think that's a word. So just 1 minute and we'll get started. Just giving it a few more seconds to allow participants to enter into the meeting. So hold tight, everyone. And we'll get started shortly. Okay, I'll make a start. So kia ora agus diaguit the corda. You're very welcome to today's State of the Science webinar series, which is being run by the New Zealand Agricultural Greenhouse Gas Research Centre. Today's very special webinar. It's being run in partnership with the Teagasc Climate Centre from Ireland. So wherever you are in the world today, we are really delighted to have you in our audience. So today's webinar is the first in a series of webinars planned, where we're planning to look and highlight research that is ongoing and relevant to both New Zealand and Ireland. So please keep an eye on our socials for more information throughout the year about upcoming webinars. Today's webinar is all about the potential to breed for climate-friendly animals. And I'd really much like to welcome Collier Isaac, who's a member of the NZAGRC's Governance Board, who will be your chair today and take you through the session. Collier has fantastic experience in the primary sector here in New Zealand, and we're extremely lucky to have him here today. So over to you, Collier, to kick the webinar off. Slán go fóill.
Thanks, Sinead. Welcome, everybody. 380 odd people registered, so there's obviously strong interest in this territory. Getting right to the point, we'll hear from 3 speakers today: Donna Berry, Peter Amer, and Suzanne Rowe. We'll just do quick introductions now, and then we'll get on with Donna as the first speaker. Donna is a senior principal investigator in quantitative genetics at Teagasc, Ireland, holding professorships at several national and international universities, as well as research. She is strongly engaged in third-level education and extension to a wide range of different stakeholders. She's also director of the Vista Milk SCFI Research Centre on AgTech with 232 staff, where the focus is on delivering fundamental and translational research in digital dairy production and processing across the whole soil-to-society food chain. Following his BAg Sci at University College Dublin, he undertook a PhD in quantitative genetics in collaboration between Teagasc, Wageningen University and University College Dublin, Ireland, followed by an MSc in bioinformatics at the University College Cork. Just scroll my page down slightly here. Working across species, dairy cattle, beef cattle, sheep, horses, his research interests are in phenotypic strategies, breeding, goal development and deployment, genetic evaluations, and genomic predictions. Of late, his interests have included beef on dairy matings, with the goal of providing information and tools to help decision-making on all things related to beef on dairy breeding. Well, it's quite a list there, Donna. Peter Amer is a world-class scientist renowned for his multifaceted approach to designing and developing effective breeding programs. He has a talent for creating practical, innovative solutions that apply to a range of complex issues, whether it be technical, commercial, or political. He's a Group Managing Director at Abacus Bio in Dunedin, leading a team of 45 international consultants. He also retains close involvement in client activities, leveraging his technical experience across animal and plant breeding programs. Global warming issues and deployment of genetic improvement in developing countries are a particular passion of Peter, or for Peter. Last but certainly not least of our speakers today is Suzanne Rowe. She is a quantitative geneticist and principal scientist at AgResearch. Suzanne leads a program breeding for low-emitting ruminants, applying a suite of technology to New Zealand flocks and herds to determine the utility of breeding for the mitigation of methane emissions. The team is currently working with Irish colleagues, hence the connection here, to determine new predictors for low methane breeding schemes, and we were recently awarded the prestigious Royal Society New Zealand Pickering Medal. As we get along, whack your questions into the questions section, and when we get to the end of the session, we will run through all those questions, because I suspect who might want to answer them, unless you specifically ask someone for an answer. It could be any one of the 3. Right, Donna, the floor is yours.
Good stuff. Right, thanks, Collier. So just checking, you can see my screen, yep. Good stuff. And you'll be glad to know that I'm not going to mention anything about the rugby over the weekend. Oops, maybe I already did it. So Sinead said that this is the first of a series. Now, I might be a little bit biased, but I think this is going to be the best talk not well, the best session of all the series, because I think breeding really is the tool to achieving sustainable gains in profitability. And my first slide is just a summary of all the reasons why I do believe that it is really an excellent strategy. So you get a close to complete adoption rate, be it sheep, beef, dairy, doesn't make a difference. You have to put the female in calf or in lamb. So you get complete adoption because it's just as easy to use a good bull or ram as it is to use a bad one. Also, the framework is in place for most countries to rapidly and seamlessly deploy any technologies that we develop, be it estimates of genetic merit for methane or other characteristics. Breeding is certainly not a snake oil. It's been proven to deliver year on year across a whole range of multiple different species. We're well familiar with the term, I guess, that the breeding is cumulative and permanent, but it is true. So the gains that you achieve, they accumulate over time and they can remain permanent. There's no need to change the farm management. Okay, be it artificial insemination or be it natural mating, there's a little bit of a genetic lag with the latter, but you don't need to do anything different. You still need to put the animal in calf or in lamb. And there's no direct additional costs. It's not like things like nutritional supplementation or vaccines. There is no cost because in the vast majority of cases, the genetically elite animals are more or less a similar price for artificial insemination compared to the genetically inferior animals. It's proven to be a sustainable process, animal breeding. And the nice thing about it, and I'll come back to this again at the very end, is the ability of breeding to improve many traits concurrently. Now, as arrogant as I am as a geneticist, I also have to state that the gains that we achieve through animal breeding are very much complementary, or what we call they stack on top of the benefits that are achieved through other strategies. I don't have time to go through all these 8 points. So what I'm going to do is just cherry pick the top 2, and then we'll get into a few numbers after that. So if we look at the deployment strategies, most countries in New Zealand and Ireland are what we're really discussing here to some extent. We already have what are called breeding indexes. So in Ireland, we have 6 breeding indexes, and you'll see the makeup of those indexes here, but don't need to worry too much about the detail. These indexes rank individual animals for their expected contribution to profitability, be it themselves or their progeny. Now, what you can see in the right-hand side, and this is some of the work we've actually worked with New Zealand, with Peter Raymer's group in Abacus Bio. On the right-hand side, see those black shadings of the relative emphasis in our dairy indexes and our beef indexes. So what we have done in the past few years, we have included what are called carbon sub-indexes of our 2 indexes. And again, that's accelerating the gains that we can achieve by not just increasing the profitability of the systems, but also their environmental sustainability. So addressing this whole sustainable food systems issue. So I guess the question we pose here today, and I'll touch on this a little bit later, is there a role for including genetic merit estimates of methane in these indexes? I certainly believe there is. Said the roadmap is already there, the extension of the system, it's already there to rapidly deploy if we had estimates of genetic merit for methane or any other environmental trait for that matter. And what we're actually working on with Peter Raymer's group, so this is a meeting between Teagasc and Abacus Bio on this co-funding between New Zealand and Ireland, is we're working on, and Peter, I think, might touch on this later, on how best to incorporate these methane-related traits into our breeding objectives or into these indexes that rank animals based on expected profitability. So the second point is that this is proven. Okay? And I'm showing you here for the different species what we call genetic trends. So that's the average genetic merit of individuals by the year of birth, moving from left to right. You can see in red, dairy, and this is no surprise. We see this in New Zealand, we see this in Ireland. The rate of genetic gain for dairying tends to be greater than what we see in most of the species. And there's a whole multitude of different reasons for that, not least the fact that the breeding objective for dairying started several years before that in beef. The rate of use of artificial insemination is far greater on average in dairy than what it would be in the beef and in the sheep sectors. Nonetheless, the rate of genetic gain is increasing. And if you look at that change over the past 20 years, in dairy, there's been over a 3 genetic standard deviation change. So a standard deviation, there's what, around 0.2% of the population is above 3 standard deviations. So imagine that today, you look at 0.2% of the population, and within 20 years, we've shifted the average of the population to that particular area. So there's a massive potential. There was a notion I was taught in university that genetic gain is slow. Complete rubbish. Genetic gain, if driven through a good breeding scheme, can actually be really rapid. And we clearly see that. Like, Collier, you're older than I am, but you know that the cows of 10 years ago are far different to what the cows or the rams or the sheep or the ewes are from today, right? So it's completely untrue that genetic gain can be slow. So for the second part, I just want to dig into a few numbers. This is real life data. I want to show you what is the variability in enteric methane emissions. And I'm just going to show you what it is for growing cattle. So these are Irish growing cattle. We measure around 750 of them annually, and this is their actual data. These are indoor fed. So like most traits like height or weight or IQ, it's what we call normally distributed. By the way, there's nothing wrong with being average. When you're average, you're actually better than half of the population. But most of us are average, but we have extremes. You can see here some animals are exhaling around 400 grams of methane, and some are exhaling around 100. So the question as geneticists we pose is, well, how much of this variability is actually due to genetics? In genetic terms, we call that a fancy word called heritability. Now, I'm not overly interested in this heritability statistic. I'm interested in the genetic variability. Interestingly, the heritability, and maybe Suzanne might talk about this later, that we see for methane emissions is around 30 to 40% in dairy, beef, and sheep. So around 30% of the variability that you would see amongst similarly managed animals is due to their genetics. But the key question for me is, what is that variability? And that's what I'm showing you here in red. There is large variability within animals for how much methane that they are producing. And just simplistically to demonstrate that, and this is real data, went into that database of that blue distribution that I showed you, and I pulled out 2 steers. They were sired by Charolais bulls from Solaire dams, and they were going through the test period at exactly the same time. So let's have a little look at them. They're a very similar age, both around 590 days. They weighed pretty similarly. They grew at probably more or less a similar rate. But you can see bull or steer B was eating around 0.5 kilogram more dry matter per day. So let's look at their methane. If you look at the bull or steer A, it was producing 205 grams of methane per day, and steer B was producing 35% more, despite the fact that they weighed the same and grew the same and were the same age. And in fact, if you actually look at the revenue from those animals, so the carcass weight, you can see that bull A, who was producing less methane and actually weighed less, obviously killed out a lot better and had a higher carcass weight. So what we have done is, again, going back to the whole rugby, we're first in the world at producing a national genetic evaluation for methane emissions in multiple breeds. So this is freely available to Irish farmers. I just threw up this list of artificial insemination bulls. That's their names. You can look at their methane and genetic merit in grams per day. So look at bull, the very top bull there, minus 9.7. So relative to the base population, that bull should be, that bull's progeny should be exhaling almost 10 grams of methane less per day. Now you might say that's small, but if you accumulate that over its lifetime and then multiply it by the national herd and compare it to the bull at the very bottom, who is producing 18 grams more of methane per day than the base, there is quite considerable differences there. So just to finish off, my last graph is to demonstrate what can we achieve. And if we look at the national inventory of enteric methanes in Ireland for a steer, it is around 117 grams of enteric methane per lifetime. And as breeders, we tend to talk about, well, we can improve things by 1% per year. But let's be really conservative here, because methane is a trait we need to measure a lot. So let's have that rate of gain. And let's see what could happen if we were to reduce methane emissions by 0.5% per year. The other thing, of course, and remember I went back to this ability of breeding to concurrently improve traits. Another really important trait for us and for you guys is age at slaughter. So if we were to improve age at slaughter by 0.5% per year, this is very simplistic. It's a linear line, but what you will see over that time period is you could reduce the methanes by selecting just on age at slaughter, or improving that, sorry, not selecting just on, selecting as a part of an index, reducing age at slaughter by 0.5% per year, or reducing methane emissions per day by 0.5% per year. You could reduce it over that time period of 25 years by 11%. However, remember I said about the key to concurrent selection? What if you were to do both concurrently? And we know we can do it. We've decades of evidence to prove that we can do it. We've done it in dairy for milk and beef, or for milk and fertility quite successfully. We could reduce methane emissions by around 22% over that time period. So just to conclude, 3 main points. The technology we're talking about here has been well proven. In New Zealand, Ireland, around the world, that we can achieve progress in the traits that we want to improve. Toolkit is already there. We know how to do this. We've been doing it for decades. We have the indexes of which we can easily slot in these estimates of genetic merit. And even if you don't like indexes, and lots of people don't like indexes, some of them don't even like genetic evaluations, you can't actually get away from it. You have to use bulls, you have to use rams. And the saying of if you by lifting the tide, all ships rise, there is actually 100% adoption for this technology. So hopefully I've convinced you that breeding is a real solution to achieving sustainable food systems. Thanks.
Awesome, Donna. Yeah, that is a really good rip through 101 why the hell we should be actually putting some pressure in the space. We've also gained some questions which we'll come to at the end, but that also means that you obviously are generating quite a lot of interest, which is fantastic. Thank you. Next speaker is Peter Amer. The floor is yours, Peter.
Okay, let me just try and sort out my slides here. Does that look okay? Got a slight concern here that I don't know how to advance the slides, but I will try. So thanks very much. Great introduction there from Donna. So my talk is focusing on the joint New Zealand-Ireland research work on breeding here. So I'm going to focus quite a bit on those results, but then try and put the results into a bit more of a global context. There we go. Okay, so I'm going to start off with some dirty comparisons. So there's no dirty Irish jokes here. I'll leave that to Donna. So basically, if we look at the efficiency gains in so automobiles or airplane travel, the gains are sort of 10% per decade and 20% per decade, which is kind of a emissions intensity argument. So aeroplanes and cars are getting a lot more fuel efficient and they're making big gains, but at the same time we're getting more overall emissions so from these industries. So we're already sort of touching on this idea of emissions intensity versus overall emissions. So that's the benchmark of what other industries are doing. So the question here is, what can we do with cattle or sheep in New Zealand and Ireland? And then let's also think about what we can learn for the other countries as well from doing this. So let's start by thinking about what breeding is doing for emissions intensity. So if you just look at the current indexes and genetic gain that's being achieved, realised genetic gain in New Zealand, at least for New Zealand dairy, sheep and beef, and Ireland for dairy and beef, we have estimates of what the gain is per decade in emissions intensity. So those vary between industries, and they largely vary based on how much genetic gain is being made in those industries. So the dairy industries are making more genetic gain. A lot of that genetic gain, say for milk yield, is causing dilution of maintenance and resulting in emissions intensity gains. So there's already quite a good story from breeding, and we don't want to disrupt these genetic gains too much. when we try and add on. So now this is a slide from some work we've done for the Global Methane Hub, and the genetics component of that. So there's quite a bit in this slide. I'm going to try and help you through it. So each bubble or circle on this slide is a measure of the total amount of methane that's coming out of various ruminant populations. So we've got global S, or GS is the global south, GN is the global north, and you can see here that there is a lot of big circles associated with particularly beef cattle in the global south. So we've got big circles and small circles. So those of the global ruminant population, how much they're contributing to methane emissions. Then if we look at the axis here, we've got the x-axis is how feasible is it to make genetic reductions in methane through breeding programs. And we'll see that we've got intensive dairy programs which make the most genetic progress out towards the right. The dairy programs, but also small ruminants in the global north are doing well. Whereas on the left, we've got large ruminant populations in developing countries which are not making so much gain. And then if you look at the y-axis, we've got high to low is whether there is an alternative. So you could argue that with the intensive dairy industries, there are potential feed additives which they can relatively easily implement into their farm management practices, which would mean that there would be maybe less of a need to apply genetics. That could be argued, but for the grazing industries, it's a lot harder to find an alternative, so they tend to be up in the top. We do have on the bottom left-hand corner the argument that indigenous dairy and beef breeds, the alternative might be just to straight out improve the management performance of those animals to make improvements before worrying about breeding for methane. So the point here is that when we talk about New Zealand and Ireland, the amount of emissions is a small part of the overall scheme. So we're talking the orange dot, the dairy pastoral orange dot between New Zealand and Ireland, we're a reasonable chunk of that. Small ruminants, global north, we're a small chunk of that blue dot. And the beef tourist global north, that's the green one, we're a relatively small chunk of that. So in the grand scheme of things, we are looking at breeding programs that have got quite good opportunities to apply genetics. But we also have to make sure that what we learn out of these programs can apply across to other industries where there's a much bigger overall amount of methane emissions. So just touching on the conundrum we have for agriculture, we've got a growing population, we've got more people and more food, but emissions intensity is not really good enough. We also need to try and reduce the overall emissions. So that's a sort of the left-hand side box. The right-hand side box here, we have these perspectives of gross emissions, which is led by national governments with targets and the need to reduce things. And then the other conundrum is, well, we want to improve overall animal production, particularly for less well-fed people in the world, so that we've got plenty of production. So there's the emissions versus the intensity perspectives. So now we need to think, well, what if we introduce methane traits into this? What would it be worth? How much value would it be adding? And we can think about this in terms of gross and intensity gains. So first point is that really we have to be careful here in thinking about how much we can actually gain, because we're already driving genetic gain for a lot of traits which drive profitability for farmers. So the point here is that as we shift selection emphasis away from those traits that drive profitability onto, say, a new methane trait, you can see the blue line is that there is an opportunity cost is that the blue line drifts down below 1 as we put more and more emphasis on the new trait. As we put the emphasis on the new trait, in this case methane, you can see there's a relatively linear gain until we get into really high emphasis, at least in the orange line. So it's tracking up. And the exciting thing here is that you actually don't have to give up a huge amount of profit in order to get quite a big gain in methane. However, we've still got to realize is that we're giving up 20% of profit here in this example, and we're only getting 60% of the maximum possible gain in methane. So we have to remember that we are making progress in a whole suite of traits, and methane will only be a part of that. And if you're wondering why this sort of magic is that you can get a whole lot in one trait and only give up a little bit in the other trait, there's some trigonometry on the right-hand side, which kind of shows you as you move across the response ellipse that you can actually get quite a bit of a gain in a secondary trait without losing too much in the primary trait or the index in this example. So now in New Zealand and Ireland, we're dealing with largely grazing industries. And there's a question around feed efficiency. Why don't we just select for feed efficiency and not worry about methane? But there is a conundrum here when it comes to thinking about the gross or total methane from an industry. So if this schematic here on the left-hand side, you can see 2 efficient cows and 1 inefficient cow on one particular farm. The big cow is the inefficient one. Okay, there is we can sort of model or think about what happens if we take that big cow on the left and the top right-hand farm, we replace that big cow with an efficient cow, we now have less total feed eaten. So if we imagine here that we just replace that inefficient cow with an efficient one, well, then we'll actually save some feed. I guess the argument here, though, is that a pastoral farmer won't just leave that saved feed sitting in the corner of the paddock. They will actually feed that to either the same ruminants or a new ruminant. So actually, if we improve feed efficiency in the pastoral situation, we may not have any material impact on the national inventory or the overall emissions. So therefore, that really creates the argument for the methane trait in this pastoral setting. The methane trait improves both emissions intensity and gross emissions. So rather than showing you a whole lot of mathematical equations and models and software programs and things, all I want to say is that we have modeled the additional benefit for the New Zealand and Ireland sheep, dairy and beef industries to get what the contribution from the methane trait might be. And that's about 1 to 3% per decade. So remember the cars and aeroplanes were 10 to 20%. The breeding using conventional dilution of maintenance for profit traits gives sort of 1 to 5%. So we've got a potential 1 to 3% per decade, which I believe is a realistic amount that we can deliver through genetics, noting that it's cumulative and permanent and all of the other benefits that Donna mentioned. So one point to make here that this does not fully stack with the emissions intensity from conventional gains. So when you start selecting for methane, we lose a little bit of the dilution of maintenance benefits because we do sacrifice some of the progress in the conventional profit traits in order to apply pressure to methane. You can also carry these calculations a little bit further to think, well, how much is it going to actually cost us to mitigate a ton of carbon dioxide equivalents? So in some situations, the opportunity cost of breeding for methane, when you work out the tons of CO2 that are reduced, that cost can be quite high. If you have a very high value breeding program, then, for example, the dairy breeding programs, which are highly efficient, okay, there's going to be a cost to farmers of diverting some of that genetic progress away from profitability and towards a methane trait. So we have to be careful with breeding with what we do. If we put too much weighting on methane, we will create this high opportunity cost to farmers. However, there are other industries where the effectiveness of current breeding programs is a bit more moderate, due to often no fault of their own, just the nature of the animals and the traits. For example, in beef cattle, if you make them grow faster, you also increase the maintenance requirements of the mature cows. So in these industries, the opportunity cost is a lot lower, and the potential from bringing in feed intake recording along with the methane recording, which at least many of the scientists in New Zealand believe is important, then we can actually add additional value through the selection for methane. We also record feed intake, and we can actually deliver quite a bit of value then to the farmers through this initiative, and effectively the opportunity cost is a lot lower. So then how do we make the prize bigger from genetics? So more and better methane recording. We need high accuracy when we're trading off, when we're throwing out a high index bull or ram because they're not good enough for methane. We want to know that we're truly confident about how good they are for methane. We can put more emphasis on methane in the trait mix, and that will depend on the commercial drivers that apply to farms, so that they're motivated to give up progress in other traits and make gains in methane. And the other thing we can do is just drive genetic gain faster. So we would all, as geneticists, like our industries to be making more genetic progress. The more overall genetic gain they can make, the more that benefit we'll get when we add methane into the mix. Just a final throwaway point is that this research in New Zealand and Ireland is key for us to understand the biology of the trait. And that will be also really important in applying learnings into other parts of the world where the methane emissions are much larger, but the breeding program is maybe less limited. The more confidence we can build on the biology and what we're actually doing, I think the better we'll be able to have a global impact. So that's the finish of me.
Awesome, Peter. Thank you. I can see the few more questions turning up here. Going to have a bit of a debate between Ireland and New Zealand, I think, between who's 1% is right or 1 to 3% per decade is right. But we can come to that in the questions. It's all about interpretation. Right, Suzanne, you're on.
Okay, how's that looking?
We can certainly see the screen. You seem to have a presenter's view, but look, carry on. Otherwise, I can see it getting to all sorts of bother.
Oh, okay.
Yeah, so don't worry, we can all see what it is.
Oh, I'm sure we
You
can.
have to see your next slide as well.
I'm sure we can switch that around. Just give me 1 second.
Your next slide, given I think what it is, will be a direct shot at Donna's comments.
Okay, how's that? Is that better?
Good. And sorted.
Yeah.
Go.
Yeah. So yeah, just wanted to remind Donna that just because the boys are struggling, you know, more than one gender exists in rugby. But we'll leave that there. Thanks for the brilliant introduction, guys. I'm going to talk a little bit more about the tools that we use to measure methane, and really, you know, think about Peter's point in the understanding of the biology and accurately measuring. And there are two ways to accurately measure. One is to measure something really accurately in a small number. The other is to measure a lot of individuals in order to build an accurate picture. And in breeding, that's generally what we do. So I've been working really hard on finding a technical measurement that indicates the methane emissions from a lot of animals, and really understanding the genetic basis and the underpinning so that we get statistical prediction at scale, so that we trust our breeding schemes and that we can go forward with implementation and impact. And in sheep, that's been working really, really well. We've come up with portable measures, and we've been working with countries across the globe, including a really close relationship with Ireland, where we're using short-term measures to measure thousands of sheep every year, and on farm within breeding schemes. And what we're seeing gratifyingly is real variation between breeding values, but really importantly, on every farm and in every flock. So every flock that we go to, we see highs and lows grazing together. And that's really important, because if you're going to ask people to select for low methane, and they can select from the rams and the bulls that they normally use, that's a much easier perspective than having to suddenly disseminate new genetics. And that's not what we think is going to happen. We think that it's a relatively benign trait. It's available to breeders to select. We just need to get on and measure it. And in sheep in New Zealand, that's been working really well. Every sheep breeder in New Zealand can get a breeding value for methane emissions, and they can sell to commercial breeders, and that can plug into calculators, and we can help them to see what impact selecting different rams have. But there's still a barrier, and the barrier is that at the moment we're measuring using chambers. We measure 168 animals a day. Our trailer weighs 3 tons. It's really quite difficult to, go up and down people's driveways, and we're really still not at the scale that we would like to be for a genetic trait. So we go back to biology. We've been breeding low methane sheep for around 15 years, and we know the differences between the extremes. So what we know from our research is that the microbiome in a low methane sheep is quite different from her high methane counterpart. So what she's doing is she's breaking down feed slightly differently. A slightly different fermentation is happening, and the energy source that's going to that animal is different. So that the mix of the carbs or volatile fatty acids that are coming from a low methane rumen into the animal is slightly different. And what that's doing is producing a bit less hydrogen, we think, which is producing less methane. At the same time, if we follow that through the animal, oh, excuse me, what we're seeing is that the makeup of the meat and the milk in the animal, so the fatty acid profile is also different. So it reflects the digestion. And this isn't different. We could feed and create this effect. But what we're seeing is actually we can breed for it too. So there are animals that are genetically predisposed to ferment feed slightly differently and produce a slightly different fatty acid profile. So there we've got indicators. We've got indicators of what's going on in the animal, and we've got potential predictors. And we started with microbiomes. What we were really interested in is the crux of the matter. If it's all happening in that rumen microbial community, then maybe we can use that rumen microbial community to really understand methane emissions. So we spent about 10 years developing protocols for sampling, for preserving samples on farms so we don't have to put them in dry ice, as for sequencing. And we've gone along with our pack chambers. So this is on farms out across New Zealand. When we turned up with the trailer to measure methane, we've also collected a rumen sample using a gastric tube. And then we have a process where we've sequenced about 1% of the microbial DNA. We're not really interested in what the microbes are or what they do. We're just interested in whether they predict methane. And we've compared predictions using the microbes and also the genetics of the host. And this is a relatively complicated table, but I just would like you to look at the bold. So what we can see here is a genetic correlation between methane that's been predicted using measures from a portable chamber and genomics, and methane that's been predicted from just using the microbes within the gut. And we see a correlation of 0.76. Now, what that's telling us is that we can go out and take a small sample from the rumen and sequence it, and we get almost the same answer as we do if we measure using methane. The difference is when we use a rumen sample, we just go up someone's driveway with a very small plastic tube, which weighs about 200 grams, not 3 tons. And we can measure hundreds a day. We're not restricted. So that's a really exciting perspective. But we still have the barrier that we're sticking a tube down an animal's throat, and that's okay with smaller animals. With cattle, it gets more difficult. And in countries where, someone might only have 2 or 3 animals, that invasive process is really, really not appealing to people. So what we've been doing is working on other ways. And one of the things that we've been working on is a very simple oral swab, just as you see on, your crime series on TV. And we were taking oral swabs and extracting DNA from those. And what we see is that if you look at these two bars here, the green is DNA that we get from microbes. So in a rumen sample, 80 to 90% of the DNA that we extract is from microbes. But in an oral swab, only about 5 to 10% is from microbes. The rest is all from the host. So what we need to decide now is that predictive enough? But also, should we in some way treat the sample so as we get rid of all the extra DNA, or should we actually take advantage of it? Because in actual fact, we spend a lot of time taking samples for host genotypes and sequencing across the world. And maybe this is a universal sample now that we can get both host DNA and microbial DNA. And we've looked at, so if you look at these correlations here, we've got methane, we've got a prediction from a rumen sample, and we've got a prediction from, we call it a buccal sample. You'll hear it called a buccal or an oral sample. And what we actually see is the prediction's the same. So we can predict from a buccal swab, and this is only a small number of animals, so there's a caveat there, but it looks as though we can predict from a buccal or an oral swab just as well as we can from using a gastric tube. So that's really exciting when we think about handling larger animals, and animal welfare and non-invasive samples. But it is still expensive. We still have to sequence something. We still have to go out and handle the animal. And one of the things that we've been doing is looking at other multidimensional predictors, other ways that we can follow the system through, look at the biology, and think about other proxies, other ways to predict methane. So we've had a study going for around 4 years. We have 560 data points. We've measured 180 ewes, and these are selection line ewes. So these are sheep, these are extremes. They've been bred for high and low methane, and we're measuring them around 6 weeks after lambing. And what we've done is we've taken 10,000 markers on each of these animals. We've taken 193,000 tags or pieces of information about their rumens. We've taken the volatile fatty acids. Those are the carbs that were coming from the rumen. We've taken a milk fatty acid profile in the milk itself, so we understand exactly how the fats are broken down. And we've also taken, with the help of LIC and a lady called Catherine Tiplady, who did a beautiful thesis on this, mid-infrared spectra. Now, those are the peaks that are used for a standard herd test. So what we've done is taken a standard herd test, we've sent our samples through the LIC labs, and we've said, please, can we have the spectra back? Because we want to see if we can predict fatty acids, and we want to see if we can predict methane. And this is a relatively complicated table, but I just want to point out a few things here. So this is the heritabilities, so the genetic variants that we see in each of the traits that we've looked at, and also the genetic relationships. So the first thing I want to point out is that there's moderate heritability. So these are these green on the diagonal. Apart from volatile fatty acids, we don't seem to be able to capture those particularly well in order to use them as a genetic trait. But interestingly, if we try and predict methane using rumen microbial profiles or milk fatty acids, they're almost exactly the same. So the relationship between that microbial fermentation that's going on and the endpoint in the milk is very, very close. So that tells us that biologically, we're following the system really, really well. And really interestingly, if we look at the mid-infrared spectra within the system, they're actually very predictive of all of the traits of the methane, of the rumen, of the fatty acids, and in particular of the milk fatty acids. So there are other ways, there are, other spectra. And to put this into perspective, a spectra might cost cents to collect. A rumen microbial profile might cost $50 to collect. So there's a big difference in the cost of some of these collection or these proxies. And finally, I just want to point out, this was a smaller study, obviously, than the 4,500 that we looked at when we looked at rumen microbial profiles. But what we see is the rumen microbial communities are not as predictive in these freshly lambed ewes as they were in other animals, in growing animals. And what we don't know is whether that's a numbers thing, or whether in actual fact, when an animal is freshly lambed or freshly calved, perhaps the rumen isn't as predictive. So it's something to think about and something to keep an eye on. But certainly we're really pleased with the proxies to date. And what we've done is have a look and see what the effectiveness would be, what the so what question. How much genetic gain would I get if I didn't measure methane, but I measured a proxy? And I'm looking here at absolute methane, and I'm looking at methane per kilogram of dry matter intake. And what I see is that the milk fatty acids, if I use the milk fatty acids, then I'd get 75% of the genetic progress if I never measured methane and for methane itself, and I'd get 66% for what we call methane yield, or methane per unit of feed intake. But again, these mid-infrared profiles are looking really indicative. Now, there's a lot of caveats around that. One of the main ones being, this is a set trial. These animals have all been fed the same. They've all been looked after the same. They're in the same stage, and what we really need to know is, would that prediction fall over? Sorry, I've got somebody outside. Would that prediction fall over if we looked at animals at different life stages? Would it fall over in different breeds? Would it fall over in different species? So we've been working with Teagasc within the New Zealand Island framework. And what we're doing is we're collecting samples in sheep, in beef cattle, and in dairy. We're using the same protocols. We're analyzing our samples in the same way. So we have a cross-species and a cross-country comparison, because we really can't believe these proxies until we see that they are robust. And to do that, that's a real team sport. We absolutely have to do that together. So we're working towards that over the next few years. And, it's a really exciting way to actually validate the predictors that we're coming up with. So this is my final slide. For us, next steps. One is to validate direct measures in cattle. So it's been really easy for us in sheep because we already had something that we could sort of compare all of these to. But at the moment in beef and dairy, that's quite a bit harder. We have systems in place that we can measure methane with, but we can't really measure large numbers of animals. So we're scaling those direct measures in cattle in order to be able to compare them or use them to validate predictors. We think the oral swabs are going to be something that's, has real utility globally. They're non-invasive. They can be analyzed in a lab anywhere in the world. So you could take a swab in Africa and, you could send it to a lab anywhere in the world. So that opens up opportunities. We need to keep thinking about better genomic profiles. So the universal predictor that we have is genomics. We can get genomics at birth. We don't have to wait till an animal's ruminating. We can actually get that at birth. So really understanding genomic profiles is something that we're working towards. And again, validating across country and across species really helps us to hone in on the parts of the genome that are really giving us prediction. We think there's a lot of potential for spectral profiles. It doesn't have to be mid-infrared. There's near-infrared. There's all sorts of spectra that can be used. And really, we're only coming to use these high-dimensional phenotypes because we're getting into an era where we can combine all of this information together. So one of the things that we're working on within this Methane Predict project is algorithms, new ways to combine all this data together and to really, revolutionize the way that we predict for breeding. I've said that we really need to be across species and across system, and it needs to have global relevance. Anything we do has to be able to be done in any corner of the world so that we can remove barriers, so that we can move forward. Methane is a global trait. So I'll leave it there. I'll thank a few people.
Righty-ho. That was awesome. I'm glad you guys sorted out which order you were going in, because that was the most logical batting order by miles, which no doubt came down to Sinead talking to you guys in the first place. We've got about 18 questions hanging around here. I'll sort of flick through a few of them earlier on, there was a question about what genetic marker is correlated causal to methane emissions, any literature or other references for that, which we might have covered subsequently, but who wants to have a quick toss at that? No hands up?
I can
Yeah, go for it.
answer that. The answer is we don't have a genetic marker. The answer is that, methane or the way that an animal emits methane has evolved over about 40 million years, and that means that it's integral to the genome, and it's many markers control it. An animal would be very vulnerable if just a single marker controlled this. So what we have to do is use genomic prediction, where we use a combination of thousands of markers rather than just one, and I don't see that changing.
Thank you. Question here, which I think goes back to Donna really, but how long till a proven commercial product is available? Because I think people are comparing, something of a rumen, a bolus or something with genetics, and I think that's obviously not a comparison. Go, Donna.
Yeah, so for beef, we've had national genetic evaluations, I'd say, for 2 years. So as I showed in the slide, farmers can go on to the Irish Cattle Breeding Federation website and see how their bulls rank for methane emissions. We have the same for sheep, and I think Suzanne and Peter, you have the same for sheep in Ireland. And for dairy, we're currently collecting data. I believe we're probably 3 to 5 years off actually having genetic evaluations for methane emissions in Irish dairy cows.
Thank you. Here's an interesting Twitter twist on things. Why is there such a large variance between these animals? Go back to I think it was your presentation again, Donna. How do you account for such things at a herd level with carbon credits? So that linkage between gain and the marketplace.
Yeah, so I think Suzanne touched on some of the biological rationale as to why there's differences. And it comes up, I think, Birgit asked a question about genetic correlations. We're lacking to estimate these correlations. A correlation is a degree of the association between traits. You need a quite considerable amount of data, right? But looking across studies, the correlations tends to be around 0.7 between feed intake and methane emissions, 0.6, 0.7, which suggests that half of that variability, and I think my coffee alluded to this as well, around half the variability in methane emissions is actually just due to differences in feed intake, right? Then when it comes back to kind of carbon credits and things like that, I believe that way we can base it on the genetic merit of the herd. So in Ireland, at least, every beef and every dairy animal, not necessarily every sheep animal, has an estimate of genetic merit for all the traits that are available. So if you had a herd of cows that were, let's pretend, minus 10 grams per day on methane emissions by 100 cows by 365 days, one could argue that that's how you could actually value the carbon or predict the carbon output, or the methane, enteric methane output of that particular herd, and then add that to the other carbon greenhouse gases.
Awesome. Question here, will the breeding methane reduction stack cleanly with using methane deducting feed supplements, or is this likely to impact weight gain too much? And so that's another twist, right? Yeah.
Yeah, I'm not sure if Rob Banks actually asked that question, because he's done a bit of work on this. And if you think I'm arrogant as a geneticist, Rob is even worse, because Rob argues with his simulation that as we reduce methane emissions through genetics, the benefit through non-genetic approaches actually is reduced because they're already producing a lot. I think we're a long way off that, Rob, sorry. So I think the both of them are complementary for sure. Now, whether there is a kind of a genotype, what we call a genotype by environment interaction, in other words, is, and we see this in medicinal uses in humans, is whether the effect of some nutritional supplement differs per animal. I believe it probably should, based on what we've seen. The extent of that, I don't know. Is it worth worrying about? I would say no at this stage.
Yeah, and I can add to that, because obviously we have extremes. So one of the things that we're really interested in is using them to answer these biological questions, and to look at the impact of feeds and inhibitors on high and low methane animals. We've done a little bit of work. We started to feed low methane feeds to the highs and the lows. Preliminary work suggests actually that the lows are responding and the highs aren't, which isn't the way round I expected it. But it almost looks as though if you breed an animal to be high methane, it's staying high methane at all costs and it's not giving it up, which was a really interesting finding. But yeah, we're working on that over the next few years.
Thank you. This might be Peter Amer. What evidence is there of economic trade-offs when selecting for methane in those schemes where selection is occurring? Is it a theoretical opportunity cost or an observed opportunity cost?
I think it's very much observed. When you bring a new trait into selection, unfortunately, we can't just get more gain in that trait. There's a limited amount of animals you can select. The easiest way I think of it is if you have a list of all your animals ranked top to bottom on profitability, and then you start having to throw out some of the top animals because they're not good enough for another trait you're considering, of course, you're going to lose genetic progress. So then it becomes, well, how bad does an animal have to be before you throw it out of the list? That's how much emphasis you put on the new trait. So there is a very clear opportunity cost of bringing new traits into indexes. And this is very complex when we have different geneticists around the world defining methane in different ways. So some of us like to focus on methane per unit of feed. Others prefer to focus on methane per day. And then we've got, the sort of the trade-offs and antagonisms become complex depending on what of those that you choose.
Yeah, and
So,
I can add to that a bit, Peter. I think it depends on the system. So in sheep, for example, we're seeing up to a 1% drop in methane a year with very little impact on profit if we're looking at sheep that are producing carcasses that are heavier or more wool. So some traits are positively correlated in a way that means you don't get as much impact, but there'll be other traits that are, the profitability is affected more. So it will depend on the system and the traits that are in the index that we're going after.
Thank you.
Can I just, Collier, I just want to just pick up on this because I think there's often a confusion around the antagonism between the methane trait and other traits versus just the overall load of adding in another trait into the index. And that's very commonly confused. And I guess the points that I'm trying to make is that the cost is just shifting your emphasis, the listing of candidates. So that cost is there. That cost is a lot worse when you have the antagonisms that Suzanne mentioned, and it's a lot less cost if you have favorable antagonisms. But I'm not convinced that there is a lot of favorable relationships between methane and other profitability traits. So it's best neutral or possibly unfavorable depending on how you define the methane trait.
Thank you. Robert Banks was obviously listening, Donna. He came back and said, I never said that we shouldn't use additives and genetics in parallel, simply that the faster we make genetic progress, the less we'll end up having to pay to mitigate methane. So, yeah. Thanks, Robert.
Poke the bear.
Someone here, is there a known genetic or epigenetic marker that indicates or correlates with higher and lower methane emissions?
We are looking at epigenetics. So we're looking at methylation status. So the answer is I don't know. I know that there is no specific genetic marker. Methylation, ask me in a year's time.
Thank you for that. Someone here, what do you think about NGTs and GMOs when it comes to animals?
For methane, I don't think they're going to be terribly useful for the animal because there's too much genetic complexity to be able to edit it. My answer is if we can edit something simply and it makes a big difference, then I'd be more than comfortable using it. Genetic modification techniques are more likely to be used in forages and on the microbes themselves.
Thank you. Question here, reduce methane through breeding, you need large numbers of phenotypes. How do you measure methane emissions? I think, Suzanne, you covered that very thoroughly in what you were talking about.
Can I just come back to that comment because it comes up a fair bit, right? And I know, Peter, you talked about the value to the industry, right? So the dairy industry in Ireland is a multi-billion euro industry. The beef industry in Ireland is a multi-billion euro industry. I can't see why we just can't get off our backsides and just phenotype the hell out of the population. We don't need a massive amount of phenotypes. So this heritability thing that we talk about, it's pretty decent, which means you don't need a huge amount of records per animal. So let's just get up there. And okay, we won't mention the company, but there's plenty of expensive pieces of equipment to measure it. like the cost of not doing this far outweighs the millions of doing this. It's millions, but it's millions relative to billions. Like, I think just do it. You know?
Anybody got a quick comment on that? Because we got to 10:29, and so I'm probably wrapping up. But any quick comments on what Donna just said? Was it
I'm
shaking? Suzanne?
I said I'm in.
Yeah. So yeah, it's not that he's not being a mad Irishman. He's on the money. Rightio. At this point, I think we better call it close. It's 10:30. From my perspective, that's been really interesting, and I think given the number of questions and the number of people attending, they've also seen it's interesting. I think a lot of people will watch the video. So in a really, really, really quick summary, yeah, Donna's saying, yeah, genetic gains there, get on with it. Peter's saying, yeah, it'll come at some sort of a cost when you've got to select a few more traits that you might have done otherwise. And Suzanne says, yeah, well, you can pretty much measure anything we really want to focus on it, but if we can't, when it comes to genetics, measure things easily, cost effectively, it becomes quite expensive. So thanks again for your presentations. Thanks again to everybody for attending, and have a good day or night, depending on which side of the planet you're on. Thank you.
Thank you.
Everyone.